Well, good morning, everybody. Welcome to this annual symposium of the Mount Sinai Children's Environmental Health Center. The title you see on the slide here. For those of you who don't know me, I'm Philip Landrigan. I'm a pediatrician, and I'm the director of the Children's Environmental Health Center, as well as the chairman of preventive medicine at Mount Sinai. So welcome. Our topic today is environmental chemicals and the developing brain. And our featured speaker, whom I'll introduce in a few minutes, is Dr. Robert Wright, who's a new member of our faculty who joined us in August from Harvard, where he had been for almost 20 years. And we were lucky enough to recruit him and his wife to Mount Sinai. He's a pediatrician, she's an internist, and his primary appointment is in our department. And for many years, he's been studying this issue of environmental chemicals and their impacts on the developing brain. If you want to put this talk this morning into a more historical perspective, a consistent theme that's run through these talks over the years has been our focus on the exquisite vulnerability of infants and children to toxic chemicals and other harmful influences in the environment. And last year, the particular focus was on endocrine disruptors, what happens if babies in the womb or children in the first months and years after birth are exposed to endocrine disruptors. Today the focus is on the brain. The two topics clearly overlap to some extent because the brain certainly has influence over the endocrine organs and the endocrine organs have influence over the brain, but still for study purposes we can dissect it out and talk about them separately. Let me begin here by taking just a few minutes before I introduce Bob Wright and summarize a bit for you where we stand generally in children's environmental health today. Some of you will have heard this before, so I'll move through it quickly, but I won't assume that you've all heard it. So first of all, why should we be concerned about the environment, its effects on children's health? I think the first reason, very compelling, is that rates of a whole series of diseases in American children are rising. I'll show you some data. So here are the data on asthma extending back to 1962 when CDC started keeping records on asthma. And you can see that there's been this steady increase in asthma in Americans of all ages, but especially among children over the past 50 years now. It's really a long, steady increase. Here in New York City, we're seeing a little flattening out. These are national data. Here in New York, we're seeing a little flattening out in the past 2 or 3 years. I think it's a combination of aggressive management of pollution and aggressive intervention by pediatricians and primary care providers who are really counseling again and again parents about not smoking around their children, about not having wall-to-wall carpeting, about removing sources of dust in the home. But still there's this steady rise in asthma rates across the country that now spans multiple decades. There's another very worrisome trend is the incredible increase in obesity. Obesity has more than tripled among American kids. In the 1970s, the percentage of American children who met the definition of obesity in terms of body mass index was 5%. Today it's 17 or 18%, so more than tripled. And then hidden within that overall trend are very strong discrepancies by race and socioeconomic status. We see that very poignantly here at Mount Sinai since we straddle the divide between East Harlem and the Upper East Side. Rates of obesity are really quite low among children in the Upper East Side, most of whom are from middle to upper socioeconomic status, and north of 96th Street, the rate is well into the 30s. It's really quite frightening when you realize that an obese child has a 75% likelihood of becoming an obese adult, and obese children are at increased risk of hypertension, increased risk of diabetes, and when they get into adult life are going to be at increased risk of heart disease. Now, some of that increase in obesity, a lot of it in fact, is due to lifestyle, the fact that children today spend more time indoors, than outdoors compared to children in the past. The fact that children today have such extraordinary access to fast foods that are full of all the wrong kinds of calories. But we're also, in our center, we're pursuing research that's exploring the question, are there certain chemicals in the environment that further tilt the balance in the wrong direction? Chemicals that have come to be known as chemical obesogens. Think of them as the opposite of a diet pill. Environmental chemicals that when they get into your body, alter metabolism, alter the set points for metabolism, and increase the risk that a child will become more obese at a given diet. We've done studies in experimental animals where we take a couple of groups of mice and we give them, they're genetically identical, and we give both groups access to exactly the same foods, but one group is dosed with whatever chemical we're concerned about. Most recently it's been DDT, and we found that the animals who are exposed to DDT, all other things being equal, nonetheless are fatter. So clearly the chemical is making a difference. And the question is, how many other chemicals have this effect? How many other chemicals are contributing to this trend in obesity? It's an active subject of research at the moment. Childhood cancer, the good news on childhood cancer, and leukemia is the most common form of childhood cancer. The good news is that the death rate is going down, and that's because our pediatricians, our oncologists have come up with just absolutely groundbreaking therapies to treat childhood cancer. 75 or 80% of children who are diagnosed with leukemia now survive the disease and go on to grow up and become adults. But any of you who've ever been on a cancer ward know that the treatments are terrible. They're very tough, and it's something you'd wish on no child or no family. And we see that there's been something like a 35-40% cumulative increase in the incidence of childhood leukemia in this country since we started keeping national records back in the early 1970s. And we know some of the factors that are causing this, radiation, certain pesticides, but we don't really have a coherent explanation for that overall increase. And we've seen a similar increase for brain cancer, which is the second most common form of childhood cancer. Then when it comes to developmental disabilities, which is much closer to the heart of today's discussion, developmental disabilities mainly involving the brain and central nervous system, it's about 15% of children in this country today who have one or another problem on this spectrum, about one child in 6 approximately. And there's a whole range of specific conditions included under the rubric of developmental disabilities: attention deficit disorder, autism, and beyond autism, the so-called autism spectrum, dyslexia, mental retardation, and then other non-specific behavioral problems. And autism now, as I'm sure many of you are familiar, has every year, it seems, every 2 years, when CDC releases their new statistics, the rate is higher. Today it affects one child in 88, according to the CDC statistics, 23% increase from just 6 years ago, 78% increase from a decade ago, and a very striking discrepancy between boys and girls in the rate of autism. And here's the current figures. There's been a lot of debate about what's causing this. How much is better recognition and better diagnosis? How much is a real increase? Those are the two explanations put out. I think the answer is it's a combination of both. There's no question that there's much more awareness of autism and autism spectrum disorder. People that a generation ago would have been called odd are now called Asperger's, things like that. So there's certainly better recognition both among the public and certainly among the medical community, the pediatric community. But you talk to anyone who does special ed or who works with children or who does clinical pediatrics, and they are unanimous in their view that the actual rate of these conditions has also gone up. It's not just better diagnosis. So the figures may be a bit inflated by better diagnosis, but there's no question that there's an increase going on here. We've been very involved in our center in trying to find the causes of that. Increases that occur this rapidly are way too rapid to be genetic in origin. Genetics changes over the centuries, over the millennia, but not over a few years or a couple of decades. So it's clearly non-genetic factors that are driving this increase. Is it nutritional? Is it medication? Is it lifestyle? Is it toxic chemical? Is it all of the above or some of the above? Probably a mix of all of the above. And we're doing prospective epidemiologic studies in our center to try to dissect out these causes. We published an editorial in Environmental Health Perspectives, which is one of the big journals in the field, just a few months ago. Luca Lambertini and I from our center, and Linda Birnbaum, who's the director of the National Institute of Environmental Health Sciences, NIEHS, the three of us co-authored this piece. We talked about the rapid rise in rates of autism. We talked about the fact that it almost certainly had to be environmental in origin, this increase. And then the pièce de résistance was that we put in the editorial a list of 10 chemicals that are our top suspects. Not that we're saying, mind you, we're not saying that these 10 chemicals are causing autism, we're not at all, but we're saying these are chemicals that have to be on a short list of suspects demanding urgent research. And this is just more data on the rates. So why have they increased? Well, we've already talked about this. Diagnosis is part of the story, but we think early exposure is a part of it. There was a very authoritative report a few years ago from the National Academy of Sciences that reached the conclusion that 3% of all learning disabilities, broadly defined, are due to toxic exposures in the environment, things like lead, mercury, pesticides. These are children who take a very heavy hit on these chemicals and are left disabled. Then they went on to opine that 25% of learning disabilities are the result of a combination of an environmental exposure and a genetic susceptibility. And I think when you think of it, a lot of human disease works this way. If you think of human disease on a spectrum, there are a few cases at one end that are purely of genetic origin, children who are adults who receive a very heavy exposure to something and get sick almost entirely as a consequence of that exposure. At the other extreme, you've got diseases that are purely genetic in origin. Sickle cell disease is a powerful example. Certain inherited conditions like glycogen storage diseases, rare diseases that the people in our Department of Genetics have focused on. And those two extremes maybe account for 5 or 10% of human disease at either end. But the 80 or 90% of all human disease, which is in the middle, is almost certainly a combination of some kind of an external exposure and an individual susceptibility. Out of 100 smokers, why do only 10% get lung cancer? Out of 1,000 people that eat a high cholesterol diet, why do only 25% get myocardial infarctions? There clearly are individual differences that play out all over the place. So this conclusion in regard to learning disabilities is not surprising and really is quite concordant with what we know about human biology. And we know especially the recurrent theme of this seminar, this symposium, is the notion that in early development there are particular windows of vulnerability when even small dose exposures to toxic chemicals can cause bad effects. The small dose exposure is too low to have any meaningful impact on an adult. We know that these diseases caused by toxic chemicals in children are very expensive. A few years ago, the number was $55 billion per year. Now, Leo Tresande, who used to work with us and is now at NYU, has done an updated analysis. He's brought in mercury, which was not included in the previous analysis, and he also brought in some updated economic figures. And his best estimate of the total annual cost of these diseases, you see it here, $76.6 billion. It's an enormous number. It's an enormous number. Even in Washington, it's a real number. And it's on the basis of this extraordinary cost of disease, plus the moral cost, plus the human cost, that we've been able to persuade the federal government and to some extent the state government in New York to support programs and research in children's environmental health. What about the environment? The environment as many of you in this room know very well, because you're involved in environmental issues, it's profoundly changed over these years. There are now 80,000 chemicals, actually closer to 85, that have been invented over the past 50 years. The Centers for Disease Control, the CDC, does rolling surveys where they go through the American population, taking blood and urine samples from a statistical probability sample of the U.S. population each year. They're finding more than 200 of these chemicals in virtually all of us. I suppose if you live deeply off the grid in northern Idaho, you might be able to escape some of these, but otherwise, if you're living in any kind of a city, if you're buying consumer products from the supermarket, you can reduce your exposure, certainly. You can shop smart, but you're not going to avoid these chemicals altogether. They're ubiquitous in our environment. It still makes sense, though, to shop smart and do what you can to cut your exposure. Eat organic, avoid phthalates, minimize use of pesticides, all the things that we talk about in this seminar. And we have handouts right outside that you can grab as you're going home that will give you a whole series of tips for things you can do in your own home to reduce your exposure, your children's exposure. And here's the real kicker. Chemicals themselves aren't bad. A lot of them make our lives better in many respects. They include new drugs, new chemotherapeutic agents, new building materials, new motor fuels, lots of things that make our lives easier and materially better. But the real kicker is that fewer than 1 in 5 of these chemicals have been tested for their toxic effects on children. So children are exposed to these materials in toys, in consumer products, in household goods. Pregnant women are exposed to the same products, and most of the chemicals go right across from the mother's circulation to her baby's circulation during pregnancy. And yet 80% of these chemicals to which pregnant women and small children are exposed are chemicals whose toxicity has never been examined. We just put them out willy-nilly into the marketplace. And we've learned before, to our chagrin and to our horror, that some of these chemicals can cause serious harm. And we really have to do better. We've been working to try to fix this situation. We've been working with the Congress, specifically with Senator Lautenberg of New Jersey, over the past few years to help him with his legislation called the Safe Chemicals Act, which is intended to remedy this terrible situation. The Europeans have remedied it in the European Union. We're behind the European Union. In fact, some of the chemical companies are now selling products in the States that they can no longer sell in Europe. They're dumping over here because of our weak legislation. Maybe the new Congress that's about to take over in January can do something about this. And the evidence is strong that these chemicals cause disease and disability. They're not benign, or at least they're not all benign. Prevention works. I'll close my opening remarks with an example I've used before, but I think it's such a brilliant example it's worth repeating. Lead in gasoline. Back in the 1970s, we used 100,000 tons of lead each year in this country as an additive to gasoline. It was added to gasoline for cars and trucks to improve engine performance, so the fuel would burn more evenly, so cars and trucks could go faster. And the lead that went into the gasoline all came out the tailpipe. It contaminated our cities. It created corridors of contamination in the countryside along the interstate highways. The average blood lead level of American children back then was around 17 or 18 micrograms, which is a level that would almost certainly call for a hospital admission today, but it was considered the norm back at that time when a lot of us were growing up. And in 1976, EPA made the decision to remove lead from gasoline. And if you look at the red line there, that's the decline in the use of gasoline from 1976 to 1980. And the vertical axis on the left shows how many thousands of tons of lead were used in gasoline each year. It went down from 100 to about 45 or so over that 5-year span. Now, it had been predicted, the white line there, that the average blood lead level, which is shown on the right vertical, might come down by a microgram or two from 17 or 18 to 15 or 16. That was the prediction, but the actuality is represented in the yellow, that the blood lead level came down by 50%. It fell down to less than 10 micrograms in 4 years. So an almost 50% decline in 4 years, which has extended out to the present, so that today the average blood level in American children is below 2 micrograms per deciliter. It's really an amazing triumph for public health, and the consequences of that were not only the 90% decline in blood level, but also an increase in IQ, so that the children, if you think your kids are smarter than you are, there's a reason why. Each decline in blood level produced a corresponding increase in IQ, and it's been estimated that the average gain in intelligence across the population is somewhere between 2.2 and 4.5 IQ points, which is very significant, and results almost in a doubling in the number of children in the population with an IQ above 130, which is clearly where we all want our kids to be, and a 50% reduction in the number of children with IQs below 70. And another aspect of this, which I find fascinating, is that greater intelligence translates to increased economic productivity across our society. As a general proposition, smarter people are more economically productive. We probably all know counterexamples, but still, that's the main thrust of it. And the economists up at Harvard have calculated that each year, somewhere between $100 billion and $300 billion are returned to the economy by virtue of the greater economic productivity of each crop of children who have been born each year since 1980. It's an amazing success story, and this is the kind of thing that makes me get up in the morning, even on a gray day like this, and say, it's worth coming into work, because if we can contribute to positive gains like this, the whole thing is worthwhile. And that's really what our center is about, is doing the research that makes these kinds of exercises in prevention and treatment possible. So that's what our center does. We have two main functions. The first is to do research, which is specifically designed to discover unrecognized environmental causes of disease. And then our second job is to translate our research findings into prevention, into treatment, into what I call evidence-based advocacy. Evidence-based advocacy when we go down to Washington, up to Albany, up to Hartford, down to Trenton, and argue before the federal regulators or the state legislatures about the need to protect children against environmental hazards. And this is a little schematic. We raise funds from generous donors as well as through federal grants and foundation grants. The funding supports research. The research turns into evidence-based advocacy, and the end product of it all is societal change. And here are a few specific gains that we've made through that paradigm. One thing that we do is we have what we call a pilot project research program. The way this works is that each year we award a series of relatively small grants, and most of them between $10,000 and $25,000, to researchers at Mount Sinai, in departments across Mount Sinai, who are interested in doing work on on issues in children's environmental health. And the goal is to give mostly young researchers this seed money, which enables them to get initial data that they can then leverage into getting grants from the National Institutes of Health or from big foundations like the Gates Foundation or the Ford Foundation. And we track the return on investment. So we put out between $100,000 and $200,000 each year in the form of these small grants. We put out $700,000 altogether over the past 5 years. And these $700,000 have yielded a return of more than $8 million in federal grants, which means that we now have this big engine of research in our center, which is in a position to make new discoveries every month and every year. And it all begins with the generosity of the people who support our center. I'll point out to you that that's a return on investment of greater than 10 to 1, which is even better than what Bernie Madoff got for his people. We've also turned our research into real action. We've been very active in seeking bans on bisphenol A, BPA. We worked with the state legislature in Connecticut to ban BPA from cash register receipts. Most of those carbonless receipts that you get at the supermarket contain BPA unless you live in Connecticut. And many high praises to Rhonda Sherwood, who was our liaison to the groups in Connecticut who led that effort, and also in Suffolk County, New York. We become an authoritative source for the media. They're calling us all the time. I was on NBC Today show just yesterday talking about carbon monoxide in schools and the need to put carbon monoxide monitors in every school. Yes, they are required in New York City, but there's only 2 states that require carbon monoxide monitors, and I think Baltimore is the only other big city that does so. So that's it's low-hanging fruit. Costs only $40 per school to put the monitors in. You can save lives and protect children's health. So let's now turn to the main topic of the day, the question of how do environmental chemicals impact the developing brain. And to discuss this in detail, I'm going to introduce Bob Wright. So let me tell you about Dr. Wright and then bring him to the podium. He's a pediatrician who went to medical school at the University of Michigan, did his pediatric training at Memorial Children's Hospital in Chicago, Northwestern University, did further training in pediatric emergency medicine at Brown University in Providence, and then moved to Boston about 19 years ago, where he completed his training in epidemiology and public health, became expert in environmental epidemiology, also developed very substantial expertise in genetics and epigenetics, so he has the tools to examine how toxic exposures in the environment interact with the human genome and the epigenome. He's done a great deal of work on lead and other toxic chemicals in the environment, examining in epidemiological studies how these chemicals cause learning disabilities in children. As I said at the beginning of the hour, we were extremely fortunate this past summer to recruit Bob to Mount Sinai from the Harvard School of Public Health and Harvard Medical School, and he's now been appointed a full professor of preventive medicine, professor of pediatrics at Mount Sinai, and it's a delight to bring Bob to the podium this morning. Bob, it's yours.
So I'm very happy to be here. This talk is meant to be sort of a big picture talk. It's meant to be a talk that sort of describes really, in some sense, what I'm doing, but also the directions that I see both my work in the field going in. And because it's a big picture talk, I have to lay a lot of foundations in order to build up to the big picture. So it's going to start with brain development, and just sort of a summary in very general terms of how the human brain develops, how chemicals can affect the processes that are involved in brain development. I'm actually going to shift a little bit to social environment and how that affects brain development. That's because I'm very interested in the way 2 different fields interact. And I think that's because we tend to study diseases and we tend to study even environmental chemicals in isolation. We try to isolate their effect, but in reality, nobody lives in a vacuum, nobody lives in a cage, so everybody's exposed to multiple chemicals and everybody's exposed to a different type of social environment, and that may actually change how toxic a chemical is. And if that changes how toxic a chemical is, that might provide clues to treatments. And that's really what I want to try to get at in this particular talk, is that there may be a way to treat toxic chemicals, which is actually something which is a little bit different in the field of environmental health. And I'm also going to talk a little bit about a relatively new science called epigenetics, which Dr. Landrigan talked about briefly as well. And epigenetics plays a big role in both brain development and how chemicals work and how the social environment works. And it sort of links these different aspects of the talk together. And at the end, I'm going to propose some ways we can take advantages of all these principles and develop treatments for environmental chemicals. So what in our environment causes neurotoxicity? Well, it's a very wide range of agents. Some of them are chemical, some of them are physical, some of them are psychological or social. With respect to chemicals, there's a fairly long list of chemicals that are relatively well accepted. Others are still somewhat disputed in terms of how toxic they are, even whether or not they're toxic. They include things like metals. I don't think anyone disputes that lead is toxic, but and methyl mercury probably is a fairly well studied and well accepted developmental toxicant. Some newer metals that are being studied right now are manganese and arsenic, and there's a whole host of organic chemicals. And organic just means that there's a carbon base to the chemical. It doesn't mean that something organic produced it, like a plant or an animal. It's just that it's a carbon-based chemical. And these include solvents, chemicals like PCPs, some pesticides like DDT. BPA and phthalates would fall in this category as well. In terms of other types of chemicals, broad classes, pesticides are very well known to be toxic, and certainly drugs of abuse such as cocaine, benzodiazepines, ketamine, and GHB are fairly well known to be toxic to the brain. So I'm going to start small and try to build big. So I'm going to start with a single cell. So this is a neuron. This is a schematic of a neuron. And really when we talk about toxicity in the brain, we're really talking about toxicity to this particular cell type. And a neuron consists of a cell body like all cells do, but it has some other features that are very, very different. It has an axon, which is where it transmits electrical signals, and it has dendrites, which are these shorter branches, which receive electrical signals. And there's myelin, which is really sort of like an insulator, so it allows that electrical signal to be carried stronger. It doesn't dissipate quite as fast because of the myelin. And then there are the connections between two different neurons, which are called synapses. All this I'll come back to. So chemicals can work at those specific areas. So they can attack the neuron itself, and that's called a neuronopathy. And there are some chemicals that specifically do this, and this typically can cause cell death. There's a chemical called MPTP. MPTP was famous in the 1980s. It's actually a synthetic analog of a narcotic, Demerol, that was sold illegally in California and caused an epidemic of Parkinson's disease, because it very specifically attacks cells in that area of the brain that's important for Parkinson's disease. It's actually used now in research after it was discovered what was going on to actually create animal models of Parkinson's disease. But it causes cell death in the substantia nigra. And then there are chemicals that attack the axon. That's that branch that sends electrical signals out from the cell. And there are some solvents that do that very specifically, such as hexane and acrylamide. So occupational workers who work with some of these solvents can sometimes get what's called an axonopathy. The axon starts to degenerate, and they can get tingling and loss of sensation. This is usually in the peripheral neurons. And then these are the types of toxicities you see in adults. And adults typically have a process called neurodegeneration. So all of us in this room are undergoing, to some degree, neurodegeneration, meaning our neurons that exist are slowly dying, some faster than others, but this is a process that goes on as you age. And it's actually the process that underlies Alzheimer's disease and Parkinson's disease and other types of dementia that occur as we age. Children have very different types of toxicity. In their toxicity, cells actually don't typically die. So it's very, very different. And I'll explain why children are more susceptible. And it actually has to do with the fact that at lower levels, these toxicants don't kill cells, but they do something else that actually causes toxicity. They might interfere with myelin formation, or they might interfere, and this is actually probably the most common way, with the transmission of signals between neurons. And so when you get exposed to a chemical, timing really matters a lot. If you get exposed during early childhood, a chemical is much more likely to produce toxic effects than if you're exposed in adulthood, particularly young adulthood, your 20s, 30s, 40s, even up to 50 and even 60. As you get older and you get exposed to a chemical, you already have some baseline neurodegeneration going on. That's actually, to some extent, normal. It's just a normal part of aging. Just The rapidity or the rate of neurodegeneration is what varies between people. It's present in virtually all of us. But if you get exposed to a chemical, it can actually accelerate that neurodegeneration. So elderly people are also vulnerable to chemicals, as well as children. But they're vulnerable in a very, very different way. And I'm going to talk about the way that children are more susceptible in this particular talk. So we know that the list of chemicals that cause toxicity is actually pretty similar between children and adults. What's an interesting observation is that even when you factor in that kids are smaller, the per kilogram dose that produces a measurable effect in a child is much smaller than it is in an adult. And that's because children's brains are developing. Our brains are largely developed. And there are processes that go on when a child's brain is developing that end when we reach adulthood. And it's those processes that are actually being interfered with these chemicals. And that's why we don't see evidence of cell death in animal studies where they expose animals during development, prenatal life, or what's the equivalent of childhood in an animal, and they do autopsies on the animal's brains. They don't see cell death. Cell death is associated with neurodegeneration. You do see cell death in Alzheimer's disease and Parkinson's disease, and even animal models of Alzheimer's disease and Parkinson's disease. But in developmental neurotoxicity, you don't see cells dying. So why is that? So how can we have toxicity, but we don't actually hurt the cell? We don't actually kill it. We don't even actually physically damage it. Well, if you think about what's going on in development, in order to become your brain has to organize and grow 100 billion neurons and 1 trillion glial cells. Glial cells are sort of structural support cells that create sort of the anatomical differences between different regions of the brain. They also play a role in the function of the brain, but they are playing more of a supportive role. The real brain function is being done by these nerve cells. And they have to organize. They have to migrate to form these regions. They have to differentiate into different types of cells. There's very different types of cells in different types of brain, in different areas of the brain. And then they have to form connections between each other so there can be communication. So that when I think about raising my arm actually goes up. That's what synapses are what's really forming that construct. And synapses are really the key to why children are so susceptible to environmental chemicals. So synapses are the connections between two neurons. So they transmit that signal from one cell to another. So an environmental stimuli causes a neuron to fire. And so an electrical signal goes down the neuron, and then it gets to the end of the neuron, it releases chemicals called neurotransmitters, which get taken up by the next neuron, which actually stimulate that next neuron to fire. And all this happens almost instantaneously. And this is just a normal part of how neurons work. So how does a brain build this enormous network of both neurons and synapses? Well, some of it is stochastic, meaning that some of it is just random. Synapses form in a developing brain just because they're next to each other. So if two cells are near each other, they're going to form a connection. They're going to form synapses. And these synapses in a child's brain, and even in a fetal brain, are made by the billions. And in some respect, they're just random. Two neurons are there, well, let's form a synapse. Let's get together. Let's hook up. So you actually see a net gain in synapses from the time a child is a fetus to about 2 years of age. There's more synapses, or there were more synapses in your brain at 2 years of age than at any other time in your life. You have far fewer synapses now than you had then. But you're likely a lot smarter now than you had then. And that's because a lot of those synapses weren't doing anything. They were just formed relatively randomly. And that's why they started to decrease. So the way we form a synaptic architecture or synaptic system that allows our brain to function is through our environmental stimuli. So we create this architecture that is relatively random initially, and that's programmed in our genetics. And then our environment takes over. So we get environmental stimuli, a neuron fires, and when a neuron fires, neurotransmitters get released into the synapse, that junction between two neurons, but other chemicals also get released, and these are often referred to as growth factors. And these growth factors are very, very important because they actually stimulate the permanence of that particular synapse. So if lots of growth factors end up in that synapse because it's repeatedly fired, the body understands that that's an important synapse. There's an environmental stimuli that keeps making it fire all the time. I need to keep that one. If there is no environmental stimuli that's making that particular synapse fire, then it lays dormant. Those growth factors don't get released, and the synapse starts to regress. So in a sense, our environment is stimulating the formation of our synaptic architecture. So our genes just lay the groundwork. It's the environment that actually determines what that particular architecture is. So synapses that produce function are repeatedly fired and they're kept. Synapses that are relatively dormant get deleted because they're excess energy. It's a waste to have them because we have to support them with proteins and energy. If we got rid of them, it would be more efficient. So that's why you have less synapses now than when you were 2 years of age. And in many ways, this actually mirrors natural selection, the process by which evolution occurs. So there's a natural selection which allows particular synapses to survive and other synapses to go away. And this is driven by environmental stimuli that release growth factors into the synapse. And this gets rid of all of our non-functional synapses because there's no growth factors being released. And this type of synapse is called a Hebb synapse. So there are certain synapses that when you're born, they have to be there. Obviously, the synapses that tell you to breathe, you don't want to have to learn how to breathe. They have to function. Those are different types of synapses. But the synapses that are involved in memory formation and learning, these are all Hebb synapses. And this is a schematic that's sort of showing this. So these are different dendrites, and this is an axon. And you're seeing that there are these connections, these synapses. And these particular synapses are repeatedly fired because there's some environmental stimuli. And so growth factors get released and they stick around. This particular synapse isn't doing much, doesn't fire very often. No growth factors get released and it starts to regress. And this is actually something that's been demonstrated experimentally. This is, I apologize that this was a study on newborn kittens. But there was a study on newborn kittens which actually demonstrates this principle very well. So the retina of your eye has receptors. It's like the end of a synapse, a neuron. Except instead of two neurons coming together, it's light that's hitting the retina, causing those neurons in your retina to fire. So you can actually block that environmental stimuli if you patch the eye. So if you take a newborn kitten and you patch one of the two eyes for a month, remember this is a newborn kitten, and then you take the patch off and you look at the retina, you'll see very few evidence of neuronal connections in that retina. If you look at the other eye, the eye that received light, you'll see thousands and thousands of neuronal connections there. And that's because in the patched eye there was no environmental stimuli, so those synapses and those neurons regressed. But in the unpatched eye they didn't. If you do the same thing, the same experiment in an adult cat, and you only patch it for a month, you won't see a change. You'll still see those neurons there. And that's because in the developing animal, this process of taking away unused synapses is very, very active. It's programmed into growing up. It's programmed into developing brains. It's not programmed into adults. It's already happened. Once you have a synaptic architecture, it's much harder to disrupt it. That's part of why chemicals tend to be less toxic in adults, at least adults who are not elderly. It's because you already have a synaptic network. It was formed when you were growing up. For a chemical to disrupt it, it really has to kill the cell. Otherwise, it really can't disrupt it. But in a developing child, it's actually those stimuli that actually form those synaptic architecture, that synaptic architecture that allows us to think properly and efficiently. So the implication is natural selection isn't just a process in evolution and how genetic variants are selected. It's actually a process that's going on at an individual level in all of us in terms of which cells survive and which don't, particularly in the human brain. So how do chemicals impact this process? Well, lead is probably the paradigm toxicant. No one disputes that lead is toxic to children. But what we do see more and more in research is that we're finding toxic effects at what are called low lead levels of 5 or less. Lead levels of 5 or less aren't high enough to kill a cell. A blood lead level of 100 that can kill cells. That's what we used to see back in the 1940s and 50s, and even as recently as the 70s and 80s before leaded gasoline and lead-based paints were removed. Sometimes children get really, really severe lead poisoning to the point where they go into a coma. If you get that much lead, cells are dying. But if you have a blood lead level of 5 or 10, cells aren't going to die. So what is lead doing in a child? Well, what it does is lead has a couple of properties which are very, very important. It interacts with a particular protein called protein kinase C, and protein kinase C is very, very important to allow a neurotransmitter to be released in a synaptic junction. So if there is lead around, it interacts with protein kinase C and it inappropriately tells that neuron to release a neurotransmitter. There was no environmental stimuli, but the neurotransmitter was released anyway. The other thing lead does, it's very similar, or has very similar properties to calcium. Calcium is very important for the electrical transmission of signals in neurons. And lead blocks calcium channels, which have to open up for that electrical signal to be transmitted from one neuron to another. So lead does two things which are kind of counter to each other. It causes neurons to release neurotransmitters, and they fire, even though there was no environmental stimuli. It also blocks normal transmission by blocking calcium channels. So it can block normal transmission of normal environmental signals, or even these abnormal ones that it's actually stimulating. So what lead is doing is it's creating noise. It's making it really hard for the brain to figure out which synapses are important and which synapses are not important. Because on top of the normal environmental stimuli that the child's experiencing, there are these neurons that are firing inappropriately, or these signals that are getting dampened by the presence of lead. So I don't know, Nate Silver has gotten a lot of press lately, and the title of his book is The Signal and the Noise, and it's a very similar principle, really, as to what's going on in brain development. Lead is generating noise instead of signal. So there's all these signals that are going on that are not actually from the environment, so they're noise. And the real signal, which is from the environment, is harder and harder to hear. And that's why you see these synaptic networks which are disrupted, and you can actually measure deficits, but no cell dies. It's the connections between the cells that are actually being formed inappropriately. So lead stimulates nerves to fire in a more random fashion, and it also inhibits neurotransmission. If you get exposed to lead as an adult, it does make it hard to think and concentrate, because this will happen to you. The difference is when you no longer get exposed, that effect goes away. In children, because their brains are developing, these synaptic networks aren't formed yet. So this noise is actually being incorporated into that synaptic network. And that's why they have less efficient brains, so to speak. So it makes it harder to think, makes them less efficient. And that's really how environmental toxicants work on the developing brain, and that's why they can actually produce toxicity without killing a cell. So as I said, lead exposure in summary introduces noise in the process of synaptic pruning. So which synapses are chosen for survival and which regress becomes relatively more random. And the net effect, particularly if it's prolonged, so the duration of exposure matters, because remember, your synaptic network is forming when you're a fetus and goes on until probably into your early 20s. So there are different times that are very, very critical, but if it's a prolonged exposure, it's going to have a bigger effect than if it's a relatively short exposure. So it's not just dose, it's also duration, but it's also timing. If you get exposed at different times of life, it's probably less of a problem. It's interesting that the time point in which children are most commonly exposed is about 2 years of age, is also the time point in which synaptic pruning is at its peak. Because remember, you have the most synapses that you'll ever have in your life at 2 years of age, and then it starts to reduce. So that process of synaptic pruning actually coincides with the timing of lead poisoning in most children, which is probably why it's particularly important not to be exposed at that particular age. Structurally, there is no damage. You won't see a difference in an MRI. You won't see a difference in a CAT scan. A child who's been lead poisoned and a child who has not been lead poisoning. But functionally, you can measure deficits. A lot of these principles are actually related to another developmental psychiatry, pediatric principle called plasticity. So the child's brain is considered to be plastic. That is, you can mold it, as opposed to the adult brain, which I suppose is more like concrete. You can't mold it. So the brain's capacity to diminish the effects of toxic insults is related to plasticity. And this occurs through the same processes as synaptic selection. So if you have a stroke, some of this is still going on. Even in adults who have strokes, they can relearn function. They can relearn how to speak. They can relearn movement. And that's because synapses are forming that didn't previously exist. Children, on rare occasions, can have strokes. It does occasionally happen. Children who have strokes tend to actually recover more function than adults. And that's because of those processes I talked about earlier, that if you use a particular synapse, you tend to keep it around. And that process is much more efficient in children than it is in adults. And that's why children can actually recover from insults much faster than adults can, and better in general. In the field of developmental psychology, there's what people call maladaptive versus adaptive plasticity. So maladaptive plasticity would be something like an exposure to lead. So this plasticity tends to evolve an inefficient synaptic network. Whereas if you had a positive social environment, that would tend to evolve a more efficient synaptic network, and that would be an example of adaptive plasticity. So this gets me into the next part of the talk, which is how the social environment may play a role in this. So stress is part of the social environment, and stress can be considered a developmental toxicant. Chronic stress impairs memory and learning capacity. There's a great deal of data on that. And it even happens in children. So how is this related to lead or environmental chemicals in general? Well, I'm going to talk about stress as if it's a non-chemical toxicant. So psychological stress activates a particular area of the brain called the hypothalamus, which is related to another area of the brain called the pituitary, which is a gland which secretes hormones, and goes to another gland called the adrenal gland, which releases cortisol. So it regulates cortisol. Cortisol is really, really important for brain development. An area of the brain that has the most cortisol receptors is the hippocampus. The hippocampus is a center of memory and learning. So most of your memories are actually stored, particularly long-term memories, in your hippocampus. So these glucocorticoid receptors, which are responsive to cortisol, actually modulate both neurogenesis and synaptogenesis, the formation of synapses. Acutely, stress tends to enhance memory formation. So you can all remember where you were probably at 9/11 or other very, very stressful events, and that's because of the stress associated with that particular event. Chronic stress tends to inhibit memory formation. People with PTSD, post-traumatic stress disorder, tend to have problems with memory formation and memory function. So why is this important? Well, there is a very interesting paradigm in the animal literature called the handling paradigm, which is very important for memory and learning. And about 20 or 30 years ago, down the road at Rockefeller Center, Bruce McEwen's lab actually discovered a lot of this. It turns out that if you pick up a pup from a rat or a mouse and you handle it, and this was actually discovered accidentally, and then you give it back to the mother, that particular pup tends to perform better on maze tests when it grows up to be an adult. So when this observation was made, Dr. McEwen's lab started looking into it more to try to figure out what was going on. Was it the stress of taking the pup away that actually promoted memory formation because perhaps it wasn't as actually positive to be handled? What it turned out to be was if you touch a pup, that rat's mother licks and grooms that rat more because she's trying to get your awful human smell off her baby. And by licking and grooming and having these interactions with its baby more, it promotes a more positive response to stress. It actually changes the programming in this hypothalamus pituitary or HPA axis. So long term, those animals actually are smarter. They learn better and they perform better on maze tests. And this licking, grooming, and another behavior called archback nursing, so this is when they're feeding the baby, it tends to cluster in family lines. So there was a question whether or not this was actually a genetic factor, was just rather than an environmental factor. And then there were some studies done about 8 years ago in a lab in McGill. This is actually Michael Meany's lab. And I apologize for the relatively French appearance of the low licking, grooming, archback nursing mother. But if you had two sets of rat mothers, one has low licking, grooming behaviors, low archback nursing behaviors, and another has very high licking, grooming, and archback nursing behaviors. And you looked at the offspring of those particular mothers, they would be very similar to their mother. They would have what's known as a fearful response to an open field test. So if you put a mouse in an open field, they will go to the sides of the wall because they get scared. But after a time, they start to realize there's no danger, they'll start exploring, which is a normal behavior. A mouse who's the offspring of a low-licking, grooming mother tends to stay next to the wall, never explores their environment. They also do worse in maze tests as they grow up. Whereas the offspring of a mouse whose mother did a lot of licking, grooming, and archback nursing tends to have a normal open field response and performs better on maze tests as they grow up. And they have very different HPA stress test response. As the amount of cortisol they make in response to a stressor is appropriate in this group, but inappropriate in this group. And this is something that may be genetic or may get what we'll call programmed. And this is the study that actually showed that this is probably programmed. So let's say you had the same group of mice, and you had the mother who had some offspring with low licking grooming, and the mother with high licking grooming and had the same offspring, her same offspring, but you switched them. It was a parent trap, or I'm not sure if that's the right analogy. But this mother raises the genetic offspring of this mother, and this mother raises the genetic offspring of this mother. So it's a cross-fostering experiment. So if it was genetic, you would expect that this would be the performance on the maze test. Because if it's genetic, it came from this mother. And this would also correlate more with this mother. But what it turns out is this is what you find. So the offspring match the mother who raised them, not the mother who was their biological mother. So these animals tend to perform just like a genetic offspring were if they were raised by this particular mother. And these offspring tend to perform just like they were the genetic offspring of this particular mother, even though they're not. And this is an example of programming. So there's a lot of attention lately to what is called fetal and childhood origins of adult disease. What happens in fetal life or in childhood life actually predicts what happens as an adult. And there's examples, particularly in the nutrition world, of nutritional factors that happen when the mom is pregnant, leading to an increased risk of hypertension or obesity in the child and the offspring when they grow up. The handling paradigm is actually an example of neuroprogramming. So events that occurred when a baby was very, very small, that were the interactions with their parents, actually predict how that child performs later on in life. That's not necessarily groundbreaking news, because we all sort of think of this intuitively, but it's something that in the world of science had to be proved. Well, it turns out that more than just proving that it happens, the mechanism is starting to be understood. And the mechanism is related to epigenetics. Epigenetics is another field which is getting a lot of attention. Epigenetics is believed to be the science that's behind programming. The fetal origin and childhood origin of adult diseases is probably being driven by epigenetics. So epigenetics is the study of changes in gene function that occur without a change in DNA sequence. It's probably the most common way by which living organisms interact with the environment. And it's actually how the handling paradigm works. So epigenetics is a very big field. So there's other examples of epigenetic mechanisms, but probably the most commonly studied is one called DNA methylation. So DNA consists of 4 base pairs: adenine, guanine, thymine, and cytosine. Of those 4, only cytosine has a potential modification called methyl. So a methyl group is just a carbon group which can be added to a cytosine chain, is a modification which is considered normal. Not all cytosines are methylated. Some are and some are not. But that methyl group is a little bit different in that it can come off or it can come on. And this is why you might have seen me with a slinky as I walked out. This is an example of DNA in this schematic. And a lot of times we think of DNA as this long string. Well, DNA is not a long string. It's actually a really tight coil. It looks like this. And those cylinders in the middle of the coil are proteins that bind DNA. They're called histones. And the way methylation works is if you want a gene to be turned on versus turned off, you have to make three-dimensional space exist so that the proteins that are necessary to replicate it and turn it on can actually attach to it in the right place. So what methylation does is it keeps DNA in a tight coil like this slinky. If you take a methyl group off, that area that you took it off opens up like this. And if it opens up like this, the other proteins that are needed to transcribe and express that particular gene can get to it. And it gets to a very specific spot. It's not getting to this area over here by my thumb, it's getting to the area that you want it to get to. And that's really what epigenetics is. It's three-dimensional biology. It's actually taking this DNA which is in a coil, opening it up at the right place so a particular gene can be turned on at the right time in response to the environment. And that's what's going on in epigenetics. And there's evidence that chemicals are changing that. In fact, epigenetics is a very important part of that synaptic pruning. There's evidence that DNA methylation changes are occurring very, very rapidly in early life. And that very rapid DNA methylation change is probably what's actually creating that synaptic, or it's what's driving that synaptic pruning and that formation of the synaptic architecture. So as I said, epigenetics plays a very important role in synaptic pruning via environmental stimuli. Epigenetic marks within neurons change with synaptic activity. And there's what's called an epigenetic opening, a synaptogenesis that's maximal during childhood. That is, it's much easier to methylate and demethylate genes that are important in this synaptic pruning process in childhood than it is later on in life. And in fact, it's believed that this is why children can learn languages much easier than adults, because they're very more plastic. That plasticity that I talked about earlier is being actually driven by epigenetics. And that ability to turn on and turn off genes appropriately so that you can learn a language is very, very easy to do when you're 5, 6 years old, and very, very hard to do when you're 30, 40, 50 years old. And that's because there's this opening that's sort of programmed. So at this particular junction in your life, if you have very positive stimuli, it can set you up for positive programming or for better performance educationally for the rest of your life. If you have very negative stimuli, it sets you up for programming that is permanent. So after you reach childhood, that ability to turn on and turn off genes in response to your environment, it's still there, but it's a lot dampened. And that's why you get these programming effects. You might have a 5, 10% inability to turn on a gene appropriately. At a functional level, on a day-to-day basis, that probably doesn't do much. But multiply that over 20, 30, 40 years, and that's why you start to see deficits later on in life. And in fact, one of my favorite hypotheses is that childhood lead poisoning is actually a risk factor for Alzheimer's disease. So you have this epigenetic programming that gets set when you're exposed to lead when you're 2 years old that causes a very small change in your ability to turn on and turn off appropriate genes. And that very small change has no functional role on a day-to-day basis, but multiply that over 50, 60 years, and then you start to see evidence of maladaptation and neurodegeneration. And that's probably, and there's some evidence in animals actually that that's actually what's happening. Sadly, if that's the case, then we're going to have an epidemic of Alzheimer's disease in about 40 or 50 years, since we had very high levels of lead in our environment 30 or 40 years ago. Well, the handling paradigm has actually been shown to be related to methylation in the glucocorticoid gene in the hippocampus. So in that study that I showed earlier with the bad mouse mom and the nice mouse mom at autopsy, when they looked at the glucocorticoid receptor gene in the neurons in the hippocampus, they found differences in DNA methylation between the two groups, which actually mimicked not the genetics of that particular mom, because this was the same study that did the cross-fostering, but whether or not the mom was a high-licking grooming mom or a low-licking grooming mom, and which mom raised that particular animal. So the epigenetics actually matched the behavior of the mother, not the genetics of the offspring. So what am I getting to with all this? Well, how does this relate to toxicology? Well, I'm very interested, as I said earlier, in the way two different fields interact, because if we can find ways to limit the toxicity of a chemical, maybe we can find treatments for that chemical. And this is actually what drives me, because I'm obviously very much in favor of prevention, and I think prevention is very, very important. But the fact is that there are thousands and thousands of children who've already been exposed. And if they've already been exposed, what are we offering them if all we offer them is prevention? And I think this is really what drives me and why I want to try to find treatments for toxicity, and why I'm interested in these interactions between social environment and toxicology, because I think some of these interactions might point the way towards treatments. And there's actually a study that demonstrates this in rats. This is a study done by Tom Guiarte, who's actually the chair of environmental health at Columbia University down the road. And Tom did a study, this is actually almost 10 years old, but he did a study of animals who were lead poisoned. And they were all lead poisoned during lactation or when mom was breastfeeding them. So it's mimicking childhood lead poisoning. And then he took the animals and he randomized them into two groups. So all these animals were lead poisoned. Half of them were raised in a standard cage, which was really social isolation. I'll show you a picture of that in a moment. And the others were raised in a different type of cage, a social stimulation cage. And they weren't raised in isolation. They were allowed to be with other animals. And they were very careful to make sure the animals didn't fight. And then they tested them on a maze study. This is the difference between a social isolation cage and a social enrichment cage. So in this particular experiment, all the animals were lead poisoned. And after they were lead poisoned, some of them lived in a cage like this. They had free access to food and water, but nothing to stimulate their environment. And the other animals were raised in a cage like this. If they got scared, they could hide. They had wheels that they could play in. They had ability to exercise. And because there were other animals in the cage, they had social interactions with the other animals. And then these are the results of the maze test. So this is their performance on working memory. So this is how fast you learn something. So less time is better. So these are the animals that didn't get any lead poisoning but lived in a social enriched cage. So they were the control animals. These are animals that lived in social isolation but did not get lead poisoned. So there's actually 4 groups. And these were the animals that were lead poisoned but were raised in a socially enriched environment. And then the last group are the animals that were lead poisoned but were raised in that socially isolated cage. And these animals do by far the worst. And the animals that lived in the social enrichment cage actually had a significantly better performance on the maze test in terms of their ability to learn than these animals. This suggests that social environment was actually treating lead because the randomization to the socially enriched environment happened after the lead poisoning had occurred. And then this is a test of retrieval of acquired memory. So the higher you are, the better the performance. And it pretty much mimics the same results, not quite as linear, but the animals that performed worse were the animals that lived in social isolation and were lead poisoned. So it's a combination of the two, which was the most toxic. So in terms of how this all interacts, so the lead and social interaction, some of how they relate. The brain is plastic. We know epigenetics mediates this plasticity. And the same properties that make children more susceptible to lead poisoning also make them more susceptible to social stress. But if we can change one aspect of it, the social stress, with positive interventions, there is some animal evidence that we could overcome some of these toxic insults. And this is important because right now we don't have a treatment for lead poisoning. At present, there's nothing. And in fact, if you look through the literature on lead poisoning, there's what I would call a dogma. And this is actually something I noticed many years ago, and it's something that I've never really believed, is that the phrase "the damage is done" is scattered all through the lead poisoning literature, meaning that children who are lead poisoned show deficits throughout life into adulthood. That's true. But if you don't do anything to intervene, why would you expect that not to occur? So perhaps it's not because lead poisoning is permanent. And I think that beginning part where I showed you that cells aren't dying. If cells aren't dying, there's no reason to think lead poisoning has to be permanent because the cells are still there. They just have to learn how to make more efficient connections between each other. And they can do that with a positive, socially stimulating environment. And I think the only reason why lead poisoning is long-term is because we don't intervene. And if we intervened, we could actually help children who've been lead poisoned. So I think the treatment for lead poisoning is a real possibility, and it's something that I'm very interested in developing. I have some ideas about actually using computer gaming software. I know sometimes people think that's crazy when I say that, but I actually think video games are the key to this. But anyway, so the mechanism of lead poisoning coupled with the biology of developmental plasticity suggests that treatment is possible. And the principle of critical developmental windows suggests that the timing is key. It's probably too late to intervene when somebody's 30 years old. But when they're in middle school or elementary school, I don't think it's too late. It's a little bit harder. To be honest, if we fix poverty, we'd probably fix a lot of these problems. But if we can intervene even cognitively, and there are some new softwares that are being developed that are actually meant as cognitive interventions. They've never been tried in lead poisoning, but one of the things that I'm very interested in doing is trying a randomized controlled trial of some of these cognitive interventions. The problem I've had, because I've tried some of these softwares, is that they're not very fun, and we're talking about children. So I actually think if we could make it fun, if we could create a video game that taught strategy, that exercised the brain with respect to working memory, that actually might be a very efficient way to treat lead poisoning. And that's something that I'm very interested in developing over the next few years. So in summary, brain development requires the formation of very efficient neuronal and synaptic networks. Environmental stimuli are signals which allow for the selection of synapses. Lead causes toxicity because it causes neurons to fire inappropriately, and it blocks normal signals that come from the environment. So it adds noise to a normal signaling process. It makes neuronal networks that are less efficient, and it causes measurable deficits even though there's no anatomical change seen in a lead-poisoned child. And social context might modify rather than confound lead poisoning. That is, another aspect of this is, for many, many years in the lead poisoning literature, there was a counter-argument that lead isn't toxic, poor children get lead poison, poor children do poorly, it's just what's called confounding. But in reality, what might be going on is synergism, so that the toxicity of lead, which I think is well established as being real, along with the toxicity of a very dysfunctional social environment, a very stressful social environment, actually may make lead more toxic. So the converse is also true. The idea that positive social interventions can reduce lead toxicity, I think, is real, but the converse is also real, that very chaotic social situations can actually make lead more toxic than it otherwise would be. But the bottom line is, what I'd really like to try to do in the next few years is bring treatments to environmental health. I think environmental health appropriately has been very focused on prevention, but finding treatments for children I think is very important, because there are a set of children that really have not been, we haven't really offered them anything, and I really think we need to not forget them. Is the kangaroo care advocated in NICU's derivative of the handling paradigm? I'm going to confess I don't know what is meant by kangaroo care. I'm not familiar with that term.
It's, when the babies are in the NICU, they want you to have as much skin-to-skin contact, so they advise
Lots of contact.
Oh.
They're not nurses, and, especially if you are close all the time, and get them.
Yes, in fact, I believe it came out of some of the research on the handling paradigm. The next question is, I've been successful at avoiding parabens, phthalates, and SLS in all topical products for my children since birth, but these are in all children's medicines. Are there any alternatives? How can I fully avoid exposing my children to these toxins? That might be a better question for Shauna, actually.
Yeah.
So how can I avoid exposing my children to parabens, phthalates? And many of these products are phthalates, parabens, and so on, are in our foods, and they're not avoidable if they're in our foods, unless you choose to use unprocessed foods. But in terms of phthalates and other personal care products and sunscreens and so on, there are a number of resources on the web you can go to. For example, Environmental Working Group has a list of actually specific personal care products. You can choose those that are paraben-free and phthalate-free, and I recommend those sources. They're really good. You can, it doesn't really help, unfortunately, to read labels because many things are not labeled. So you can actually advocate for better labeling.
Yeah, I'll take the next one. Okay, 3 or 4 different questions asked about chemicals in vaccines, mercury and other chemicals in vaccines. So this has been a topic of great controversy, as I'm sure you all know, and it's also a question that has received very, very extensive study. So there have been about a dozen really high-quality studies that have looked at this issue of whether or not there's a connection between vaccines and learning disabilities, specifically autism. There have been studies in this country, in Europe, in Britain, in Japan. Not a single one of these studies has found any connection between vaccines and autism, and not a single one of the studies has found a connection between any component of vaccines and autism. I think one of the most informative studies is one that comes out of Japan from the district of Yokohama, the port city near Tokyo. They were concerned a decade ago about rising rates of autism. They have a very centrally organized healthcare system over there, much more so than here. And so in that district, in that province, they basically stopped childhood immunization, the MMR, the measles, mumps, rubella vaccine, stopped for a period of about 2 years. Well, it turned out that during those 2 years, rates of autism continued to rise in unvaccinated children. I think that's an extremely telling example, but there have also been superb studies from Europe where they have very good population registries, and they've found no connection. I know that a lot of parents, I'm sure some parents in this room, have held off getting their children vaccinated, or they've gone to alternative vaccine schedules, such as some people have advocated. I must tell you, as a pediatrician, I'm very worried about that. Certainly people are entitled to have their opinions, but what we're seeing as a consequence of failure to vaccinate is resurgence of disease. In the state of Washington, where there's a very liberal legislation which allows parents to opt out of vaccine programs for what's called personal choice, they've had a big epidemic of whooping cough within the past year with fatalities. Whooping cough pertussis was a disease that was alive and well when I was a kid too many years ago, that had been virtually eradicated from this country and is now coming back. We've also, within the last year or two, had epidemics of measles and German measles in communities where a child typically a child who'd been traveling overseas, brought the virus back into a community where there were large numbers of unvaccinated children, and then the disease goes through the community. And a lot of young parents today haven't seen these diseases because of the triumph of vaccines. But I must tell you, from having worked a lot myself with measles, that it's a dreadful disease. It causes blindness in a certain percentage of kids, deafness as a long-term complication, and 1 out of every 500 or so children with measles actually gets measles infection of the brain, which can do everything that lead poisoning does and worse. So I know it's a subject of ongoing vigorous debate among the public, but I think it's fair to say in the scientific community the question is settled, and there just is no connection. Okay.
If chemicals are a problem because of synergistic effects, how can you study and figure this out? How can you replicate multiple chemical exposures in a lab? And how do you deal with the fact that chemicals are more toxic when combined? This is a great question. This is really at the heart of what NIH is actually trying to do, because there is a growing recognition that nobody gets exposed to one chemical. It is obviously a good recognition that this is the case. I think the counter argument is more that we'll never really understand chemical toxicity unless we start trying to look at multiple chemicals, because the reality is none of us are exposed to one chemical alone. So as long as we study everything in terms of one chemical at a time, I don't think we're ever going to really understand it. And it is a very, very difficult question. It's a relatively new field, the idea of studying chemical mixtures, but it's a field which is actually probably going to be the norm in a few years, because I do think there is a growing recognition that it's unrealistic to think that an animal study in which an animal is exposed to one chemical alone actually represents what's going on in real life, in humans, or even a study in humans, we only measure one chemical, just doesn't represent the reality. And I think that's just a tough question that I think people are starting to address.
I wanted to add to that there is a very nice study by the Environmental Working Group where they measured cord blood and found that on average there were 200 chemicals in the baby's cord blood at the time of birth, and the term pre-polluted has been used.
Okay, somebody asked a question here, where do children today get lead? There's a couple of sources. The big one is still, as it has been in this country for a century, is lead paint. There's still many hundreds of thousands of homes and apartments in this country, mainly homes and apartments that were built before 1977 when the use of lead paint became illegal, that still contain lead paint. And when the lead paint is in an old, deteriorated, poorly maintained home where the lead paint breaks up, flakes off, contributes to the dust on the floor, it's that combination of old lead paint plus poor maintenance that's especially deadly. And sad to say that the great majority of those cases where you have that convergence of factors is in poor communities. There's a lead belt in the Central and South Bronx. There's another one in Brooklyn and Bushwick. Most of the public housing in New York does not have lead paint, unfortunately. But I also want to emphasize, though, that lead paint is not just a disease of poor, disadvantaged kids. About 15% of cases of lead poisoning in New York each year are associated with home renovations. And you have families that decide to sand down some old paint and going to paint it over. They don't realize that when they sand down the old paint that they're creating a dust. It's even more dangerous if they apply a torch to the old paint, because a torch or a heat gun, because that creates a lead fume where the lead vapor gets into the air and you can inhale it, which is exquisitely toxic, terrible stuff. So, we've seen cases of this. The worst scenario is pregnant families, young family, they're pregnant, they decide they're going to prepare a room for the new baby, so they sand down the old paint. And we've had cases of lead poisoning in pregnant moms, which is, as you can imagine, just a disaster. Then beyond lead paint, there are other weird sources. You read about them in the newspaper from time to time. Trinkets that come with sport shoes. There was a child that died from having swallowed a little trinket that was made 100% of lead and it stuck in her GI tract. Some families from Asia, from South Asia, use cosmetic products like an eyeshadow that's made of lead. Occasionally there's some pottery that comes in from overseas that has lead in the glaze. So be careful what you drink your coffee out of. And that's where lead is from today. Bob?
Are there other ways, like social interactions, to do cognitive treatment instead of video games?
Probably.
Well, I should clarify, when I say video games, I wouldn't advocate really violent games. I know how popular they are, but I do think when you're talking about a child, it's very important that it be something that it's fun, because if it's not fun, they're going to drop it. And that's why I'm very interested in developing video games for cognitive therapy, because I do think that there's opportunities there that are relatively unexplored. They're actually being explored, actually commercially. There are some softwares out there. I think you see them advertised that say that they can train your brain. And I think if we can tailor them to young children, and I would argue the younger the better, because as I said, I think there are critical windows at which these interventions will be more effective than at other time points. But I do think, I personally think video games is probably the most likely to be successful. I'm not arguing against social interactions. I just think that as both a pediatrician and a parent, I think I could probably get my kids to do video games. And I think they could be tailored in a way that would be beneficial to their brain development.
Bob, can I take my prerogative and ask you a question?
Sure.
Sure. So I'm thinking about licking and grooming and what would be the analog for us. And I'm wondering whether women who breastfeed, whether breastfeeding modifies the effect of lead, whether it does help.
That's a great question. I don't know of any data on that. And certainly breast milk can be a source of lead. There's some studies on that. But whether or not it modifies it, I'm not aware of any literature.
studies do.
Yeah, I've not seen a study that looked at it that way.
Okay, somebody's thinking globally and asks, what are the effects of climate change on children's health? We've actually had a little project in our department looking at that. Dr. Perry Sheffield, who's one of our pediatricians, has been leading the effort. And the answer is that there are a whole range of effects that climate change is having and is going to continue to have on the health of children around the world, including here. One effect that's already been documented very carefully is that climate change has resulted in various diseases having a wider range than they had before, especially the diseases that are carried by mosquitoes, like malaria, like dengue. As the world gets warmer, as various regions of the world get more humid, the range of those diseases is spreading. A lot of people are concerned, for example, that dengue, which is a viral disease carried by mosquitoes, is going to become endemic in Florida and maybe along the Louisiana Gulf Coast within the not too distant future as that region continues to get warmer. There have been cases of locally transmitted dengue reported by the CDC in Florida just in the last year or so. So the infectious diseases are one realm. A second area where climate change affects children is heat, especially in hot parts of the world like India. There are some cities in India where you have 3 or 4 months now where the temperature doesn't fall below 110 degrees during the summer months, and there's no way that can be good for children. It certainly impairs their energy level. It impairs their ability to learn in school, because you can figure that those are the areas of the world where there's not much in the way of air conditioning or other ways to cool off. Then, of course, rising sea levels. We all saw the impact of Hurricane Sandy just a month ago, and children living in those parts of Rockaway and those parts of Queens like the Rockaways, parts of Brooklyn, parts of Staten Island, the Jersey Shore that were heavily hit by the floods. Those children's lives have certainly been upended. At the very least, they were subjected to stress. School was disrupted, and they're at least at risk of being exposed to things like mold that are growing everywhere in the aftermath of the storm. So those are a few of the questions. There's a lot we don't know, and it remains an area of active study.
I'd just like to add that pregnant women do not do well with high heat. You know, if you've been pregnant, that a hot tub is counterindicated, sauna and so on. And so pregnant women and their fetuses are at particular risk. Heat is a teratogen. So this is my concern, one of my concerns with global warming.
This question is, how effective is chelation therapy in reducing the level of heavy metals in the body? Well, chelation therapy will reduce the level of heavy metals in the body. However, there's no evidence that will reverse lead or even mercury toxicity. So it's not something that I would advocate. There was a large randomized trial of chelation therapy that was sponsored by the NIH in the late 1990s called the TLC study. And they took children who had blood lead levels between 20 and 45 micrograms per deciliter, and half of them got chelation, half did not. And they did neurocognitive testing on the two groups after the treatment, and they didn't find any differences in terms of their IQ or their learning or behavior. One of the caveats that I often bring up when families ask is, when you look at some subsequent studies that came out of that, even though they didn't perform any differently in terms of their IQ and cognitive tests, the kids who received chelation were a centimeter shorter than the kids who didn't. I suppose maybe I'm more sensitive to the height issue than others, but it does suggest that something bad might have been happening. So chelation agents are not specific. So There are minerals that we all need in our body. They're normal vitamins. Iron obviously is a mineral, but selenium, many others are actually minerals that are essential nutrients, and these chelation agents can actually chelate them. So it is possible that not only did it not help the IQ, but it caused some deficits in growth that actually may have been bad for them. So I think in the absence of any data showing that it improves the health outcome, I would be very hesitant to ever advocate chelation therapy, except with very high lead levels where it's potentially life-threatening.
Or high mercury.
Or mercury.
Okay, a question here about hydraulic fracking, right, for extraction of natural gas. The person writes, have there been studies undertaken to address the staggering number of chemicals found in the hydraulic fracturing process now in 34 states? So I think the short answer is there's an awful lot we don't know about hydraulic fracturing, which is why there's so much concern about it. The companies that are doing the procedure have been extremely reluctant to reveal the secret sauce that's in that liquid that they inject into the ground. State regulators have not been very hard-nosed up until now in terms of demanding that companies release this information. Some of you are aware, I know that a few people in this room are actively engaged in the current effort up in Albany to get a moratorium on fracking in New York State, or at least in certain areas of New York State, until we know more about the potential risks. I have lent my name to that effort. I was publicly quoted a few days ago in a release up there. I think that going ahead and doing hydraulic fracturing without a keen appreciation of the potential consequences is foolhardy, and we may be setting the stage for environmental degradation and health effects that could last for decades.
If you have a child diagnosed with ADHD, what actions do you recommend regarding their environment?
No, I don't.
Is it really too late?
The ones I turned over and done.
I would never say that it's too late. Certainly it's not good for a child with any kind of neuropsychiatric disease to be exposed to lead or any other potentially toxic environmental chemical. And while it may not specifically cause the disease, it can certainly make the disease worse, so even after the fact. So certainly reducing exposure to any chemical would be a good idea, any potentially toxic chemical would be a good idea. It's a hard question to answer because how you get exposed actually varies by different ages. Children have different behaviors at different ages. A lot of hand-to-mouth behavior up until about age 4. So things that are in house dust can actually enter the body up to about age 4 to 5, and then children stop doing that. So it's less of an issue for a 7 or 8-year-old in terms of house dust being contaminated with lead than it is with a 3 to 4-year-old. So you might want, so it sort of has to be tailored to the individual child. So that's why that question is actually a little bit hard to answer.
Okay, here's a question from somebody who likes fish. They want to know what are the benefits of the omega-3 fatty acids that are found in fish, especially high levels in salmon and other fish in that family, like trout and electric char. So it turns out that omega-3 fatty acids have a whole series of health benefits. They help brain development, so it's good for pregnant women to eat fish that have high levels of omega-3 fatty acids. For adults, they have benefits for heart health. They fight the formation of cholesterol. But there's a big caveat here, and the big caveat is that certain species of fish, not all, but some species of fish have high levels of mercury, which accumulates in fish because what happens is coal-burning power plants burn coal. All coal contains a little bit of mercury, so when they burn the coal, the mercury vapor goes up the stack. The mercury droplets, microscopic droplets, travel through the atmosphere. Eventually they fall out of the atmosphere, they land in rivers and lakes and the ocean. In the marine environment, it gets converted to a very toxic form of mercury called methylmercury, and that methylmercury builds up in certain species of fish to reach very high levels. Also, some species of fish have high levels of PCB. You wouldn't want to eat striped bass out of the Hudson River because it's going to have lots of PCB in it. And it tends to be the same species of fish the same species that accumulate the mercury accumulate the PCB, because basically it's the predatory species of fish that build up the high levels of both these two toxic chemicals. So how do you thread the needle here? How do you get it right? The answer is to understand that not all fish are the same. Some species of fish are high in omega-3, some are low, some are high in mercury and PCBs, others are low. So you have to shop smart. And rather than try to give you a list here from memory, I'll recommend a couple of very good websites that are out there.
They have a little handout on that in
There's
the
a
packet. Yeah.
Oh, out in your packet, good. So take instead of me giving you the websites, then just take one of the packets as you leave if you haven't already done so, and it gives this information. You can take it with you to the market when you're buying fish. The general rule of thumb, though, is the big carnivorous species like tuna, swordfish, striped bass, bluefish if you're a fisherman. Those are the ones that are high in the toxics, and the ones that are low in the toxics are the fish that basically have a vegetarian diet: flounder, sole, tilapia, catfish, things like that. All right. Well?
Is there any research that connects the early success of transcendental meditation, as noted with PTSD, and potential treatments for childhood toxicity and detoxification? I'm not familiar with that literature. I think in a child it might be difficult to get them to practice transcendental meditation, but I love that as an idea for, say, an adult who had a history of lead poisoning or occupational lead poisoning. Yeah, I think a motivated adult, I think it could potentially work. I don't want to, I know I talked about critical developmental windows, and that timing is very important in terms of both toxicity of a chemical and for treatment, but I don't want to imply that there's nothing you can ever do once you're an adult and you've had this history. I do think it's just going to be harder. It doesn't necessarily mean that there can't be success with other interventions, and I think this is a very interesting idea, actually, transcendental meditation for lead poisoning. I think it'd probably more likely be successful in an adult, though.
There's a question on flame retardants, which is a very important class of chemicals, PBDEs, which are, there's been a lot of press. If you want to read about that, there's a special in the Chicago Tribune covering a number of new studies. And so the question is, what actions are being taken to ameliorate negative effects? Well, one action is to get the PBDs out of our sodas and our cushions. And there's legislation in some states, I know in California, I don't know about New York State, but there's a lot of resistance from the manufacturers to getting these out. They're not needed, they're not actually, they don't improve the safety of the products. So if you want to do something active, want to read about PBDEs, they're definitely toxicants. They're related to thyroid cancer. I don't know if Bob wants to say more about PBDEs. But
Learning disabilities.
And PBDEs and learning disabilities? Yeah. To be honest, I'm not familiar with that.
Okay, this paper came out from our sister shop at Columbia last year. Julie Herbstman found that babies exposed in the womb to brominated flame retardants had lower IQs when they were old enough to be tested for IQ.
So it's an area to work in for those activists in the group to get legislation to remove them.
All right, it's coming up to about 25 after 11. We're going to try to close at 11:30. So I'm going to do one last question here and then close it down. So the last question I want to take is somebody asks about organophosphate pesticides and an organic diet. What are the benefits of eating organic? So there's been a lot of press about the pros and cons of eating organic. Recently, there was a report from Stanford University that claimed that there was no benefit of eating an organic diet. I'm honestly not sure where those people are coming from, because they're flying in the face of the evidence. The evidence is this. There have been several very good studies, one at CDC, another one at the University of Washington in Seattle, a third from Harvard, where people they've used various strategies for comparing families that ate mostly organic food versus those who ate mostly so-called conventional food. Conventional means pesticide treated, right? And what they found was that the families who ate mainly organic had 90% less pesticide, less organophosphate pesticide in their bodies, in their blood, in their urine. The good news in those studies is that the washout of these chemicals from the human body is rather quick. Unlike DDT, the old pesticides from 40 years ago that would basically get in the human body and stay for decades, the organophosphates go through quickly, and they found one of these studies did what's called a crossover design, where you had two groups of people, and in the first week, one group ate organic, the other group ate conventional. Then over the weekend they switched, and they collected urine from both groups in both weeks. They found all the time that the people who ate organic had much higher levels of pesticides in their body than people, sorry, got it backwards. People who ate conventional had much higher levels of pesticides in their body than people who ate organic. But after the weekend, it washed out real quickly. So that's good. It means that there's hope, and it means that you can eat organic as much as you can, and you'll benefit from it. Are there more vitamins and minerals in organic food? I don't know. But there are certainly many fewer pesticides.
And they taste better.
And they taste better. And they have, if you're thinking about global warming, local, more or less organically grown food has a lot smaller carbon footprint than lettuce that comes from California. It doesn't have to get shipped across the continent. All right, so let me, now as we approach 11:30, let me first of all thank our speakers, Bob Wright, Shanna Swan. Let me thank all of you for being here, for spending the morning with us. Thanks to those of you who have supported our center morally, spiritually, financially over the years. We wouldn't be able to do the work without you. Again, I want to thank Rhonda Sherwood and Tanya Murphy and Wendy Mendel and John Peel and any other members of our board who are here. Jackie Leitzes from our development office, who has been deeply involved in the planning, and most of all, Katie Southwick, who is standing over here in the doorway.